Good morning, everybody. In my first lecture, I would like to talk about two AFM methods, which is high-speed AFM and topography and recognition imaging. So, high-speed atomic force microscopy, what I'm talking about is the high-speed AFM developed by Toshio Ando. And I think I told already about his motivation of developing a high-speed AFM, which is that he wanted to see the myosin walking on the actin filaments. So the first paper of this development came out 2001, and then the paper with the myosin came out 2010, published in Nature. And of course, the advantage of high-speed AFM is that it's the only really high resolution, I mean, what is high resolution, but really nanometer resolution technique that can be used in physiological conditions. Electron microscopy has made a lot of progress. X-ray crystallography, of course, where you can also get three-dimensional information. But there you have to dry the sample, freeze dry. Then of course there's NMR spectroscopy. Also optical microscopy or optical nanoscopy you can get, but not really a resolution of 1 nanometer, more positional accuracy, so there's some difference. But the true resolution you can only get with AFM. And so basically, so the advantage is that you can image under physiological conditions, but traditionally AFM is a slow technique because you have to scan across the surface. Yeah? It's not like with optical microscopy or EM that you get a picture in once. But this is now what high-speed AFM overcame. So also the time resolution is high now. And the question is how you can increase the time resolution. And this is what I want to report about the strategies of Toshio Ando. And this is the paper I was talking about, so that you can see that the myosin, walking on the actin filament. Yeah. So that is really kind of spectacular. You can see that you can really see the walking process. I also want to mention, as we discussed it yesterday, there was a spectacular optical microscopy study with myosin, where they put two labels on the legs of the myosin, but asymmetrically, and then they track the distances, because the question is how is the myosin molecule walking? Yeah? Is it basically, I hope I can do it with all cable, is it walking like this, yeah? Or is it walking hand over hand, which is like this? Yeah? And in this fluorescence method, they could detect that that the myosin is walking hand over hand. Yeah, so like turning, that was already resolved with. But to really visualize it, this is of course you cannot do with optical microscopy, because the resolution is not 1 nanometer, just the positional accuracy, so you don't see the whole structure. But that can be done with the high-speed AFM. And so this paper was published in Nature 2010. And I also would like to mention not only Toshio Ando, but also Noriyuki Kodera, because a lot of these developments were done in the PhD thesis of Noriyuki Kodera. So maybe this gives you also some motivation for your PhD thesis. Okay, so I mean, in principle, this high-speed AFM is a tapping mode AFM. Yeah? So it's and it works like a tapping mode, and you know that what is held constant in tapping mode is the oscillating amplitude. Yeah? So of course, if you oscillate the cantilever, and if you touch the surface, the amplitude is being reduced, and this amplitude reduction is held constant in a feedback loop. So this is how tapping mode works, and that is not different with the high-speed AFM. But there's just some special developments making everything faster. So you have topography image, that is again how you scan across the surface, and if you hold the amplitude constant and there's an object, the cantilever is lifted, yeah, to keep the amplitude constant. And what you detect is the Z movement of the cantilever, and that gives you the topography image. So this is just like in normal tapping mode. Yeah. So if we want to make everything fast, then we have to consider all delays that come into account during detection. So this is the input signal, what you get from, well, of course, again, we have this laser beam detection, so that's the signal that we get. And this is how it's processed, and this is how it comes to the output. And every single step takes a little bit of time. Yeah. So first of all, cantilever response, then optical beam deflection detector. Yeah. So everything uses up some time. Then you have to detect the amplitude. So there's the amplitude detector. The PID controller, yeah, which provides the feedback. So this also, of course, takes some time. Then a device that drives the piezo, and then also the scanner. Yeah, this is also, so with each step of the analysis, it consumes a little bit of time, and there's some delay. Yeah, and this is what has to be made fast. So the question, how fast should it be? Let's consider the following. So let's say we have an image of 100 times 100 nanometers, and we scan with 100 lines. And the aim is to achieve a time resolution of 50 milliseconds. Yeah? And okay, I want to emphasize also, I'm reporting a paper here. Yeah? And this is the paper of Toshio Ando, Progress Surface Science in 2008. Yeah? So if you are interested in, I just can give you a short overview in all the details. You're encouraged to read this paper. And so this was a publication in 2008, 50 milliseconds, which was the limit at this time. Now the limit is at 20 milliseconds, and the aim is to push it even further to 5 milliseconds with some more features. So it's a little bit of not fully up to date anymore. Yeah? So 50 milliseconds. Okay, and then let's see what do we have on our surface. We have proteins, and let's consider the dimension of a protein is in the range of 10 nanometers. Yeah, so you basically have something like oscillation of from the topography signal of 10 nanometers, and let's just for simplicity consider it's a regular surface. Okay. So then the frequency is then the velocity divided by this kind of this oscillation, full oscillation duration, which is 10 nanometer, and that is then of course also the same as 2 times, because you're scanning forth and back. Yeah, that's the factor of 2. Then a length of 100 nanometers. And then you scan it 100 times. Yeah? So this is all the time you need, 100 nanometers, and you scan it 100 times. And then the kind of wavelength of 10 nanometers and the time of 50 milliseconds, and then we reach a frequency of 40 kilohertz. Okay? So this is kind of the image frequency. So what you need is a bandwidth of that is higher than 40 kilohertz, yeah, of all your detection and electronical system that you are able to image in this with this time resolution. And this is now the times that come from. So this is the time delay from the cantilever, yeah, because of the mechanical properties of the cantilever. Then amplitude detector, and then here we have the scanner. Yeah, that is the time delay of the scanner, and some other time delays. Yeah, and Toshio put in a factor of 1/8, And this is a kind of security factor. Yeah? So basically, because it's an upper limit, and to avoid overshoot, that is a kind of experimental safety factor, if you want to like this. This is how he describes it in the paper. So cantilever response, reading time of the amplitude, C-scanner response. So these are the important time delays. So how is the cantilever response is given by the quality factor of the cantilever divided by pi times the resonance frequency of the cantilever. Okay? Amplitude detection, let's say we need half an oscillation to determine the amplitude safely. Okay. Then the delay, the time for detecting the amplitude safely is 1/2 the resonance frequency of the cantilever. Okay. So what is clear? If you want to reach short times, then the resonance frequency of the cantilever has to be as high as possible. Okay. But at the same time, the spring constant should be low, yeah, because you wanna be sensitive. You're detecting biological samples. It's easy to increase the resonance frequency. You just make the cantilever stiff, but this is what you don't want. Okay? And this is the spring constant of the cantilever, and that is also the factor that comes into game for a rectangular cantilever, because we don't have a point mass. Yeah? And this is also the correction factor for this. So what we have to do is we have to reduce the mass. Yeah? Otherwise we cannot be sensitive, and we cannot increase the resonance frequency. And this is now the challenge for the etching technology. Okay? And so the cantilevers that are now standard for high-speed AFM, they have a length of 7 microns. Yeah? I mean, usual conventional cantilevers have a length of 50 to 100 microns. They have a width of 2 microns and a thickness of 90 nanometers, so about 100 nanometers. And in particular, these 100 nanometers are the challenge, yeah? Because the cantilevers are very thin. Yeah? So conventional cantilevers have a thickness of 500 nanometers. Okay? And this leads into a resonance frequency of 1.2 megahertz. Yeah? And a spring constant of 200 piconewton per nanometer. And this is the challenge. But then, of course, also the challenge is you have to detect the optical signal. Yeah? This means you have to improve the optical system, because conventionally the focus of the laser beam is something in the range of 10 microns, and now you need to focus it down to 2 microns. So that was the other challenge to improve the optical detection system. And to come now from the 50 milliseconds to the 5 milliseconds, maybe also just shortly tell it, is that with even shorter cantilevers, yeah, that are being produced with 2 to 3 micrometers, yeah, that is one thing. And then the other thing is to scan only into one direction, record only into one direction, and then fastly go back. Yeah. And then an additional kind of parachuting system, which is also embedded, because if you scan fast, the feedback has difficulties to follow. And so you can go out of feedback, but then the system doesn't know what to do. And there's a parachuting function in there that the amplitude is increased instantaneously until it hits the surface again, and then the system has information what to do. Yeah? So this is also a parachuting system which should be in there. Okay, and this is how it looks. Yeah, as I mentioned, there is the optical detection system that was developed here. You can see that here. And Well, also the scanner, so conventional scanner don't have this high bandwidth. And so this is the scanner, what was developed here. You see the scheme here, to suppress inertial forces and also increase the resonance frequency. So resonance frequency, and it's a very small scanner, of course. Yeah, otherwise you cannot have this high, and that means also that the sample plate is small. So what is scanned here? You can see that here it's a maybe a little bit unusual configuration. The cantilever is upside down. Yeah, and this is the sample, and the scanner is very small. This means that your sample plate is just something like 4 millimeters. Yeah, so you can only and also the scan range of the scanner is something like 300 nanometers. Yeah. So that is the way to increase the resonance frequency. And then also an active damping. Okay, so this is roughly the configuration of the scanner. Yeah, but
so
as
it's like truly upside down, or it's just the representation?
sorry?
The sample is up and cantilever is down, like as you showed in the scheme, or
Yeah, yeah,
it's like
this. Yeah,
oh, it is like this.
it's like this. Yeah. Uh-huh.
Only for the objective lens to focus the beam sharply.
Exactly. Yeah. Yeah. Okay. All right. So this gives now everything. 120 kilo. This now gives the system a bandwidth of 120 kilohertz. So that is the standard now. Okay. So kind of 10 to 30 frames per second. Resolution is better than 1 nanometer. Of course, what is also the advantage, you don't have so much drift problems because the scanner drift is slower than the scanning rate. And the scan range is something like 1 square micron, and the C 200 nanometer. And it also has some additional compounds like UV illumination for caged compounds, a syringe, that you can add ligands, for instance. Okay, and now a few examples that have been done. So actually mainly in our lab now. So this is antibodies. That was our first project with Toshiando, and it was actually started in Toshiando's lab. Yeah, so Johannes Breiner went there, and then it was finally published 2014. And that is what we saw. So the aim was to follow kinetically binding of antibodies to membranes. And then I met Johannes there in Kanazawa, and he said, our antibodies are walking. Yeah. I said, what? And this is what you see here. So basically, you can see that here the antibodies are not stable; they are walking. Here, purple membranes, it's a slow walking process. S layer, they are walking very fast, and on the strep avidin layer, they are not walking. But you can see that the direction is all the same of the antibodies. So why is that the case? The antibody has a twofold symmetry. Antibody consists of the Fc and the 2 FAB arm, and has a twofold symmetry. And these membranes are kind of two-dimensional protein crystals. Yeah? So the antigens are fixed. Yeah? It's kind of a crystal. Purple membrane is, for instance, in retina. S layer is a bacterial layer. It's the outer membrane of bacteria. And so the purple membrane has a threefold symmetry. The S layer has a fourfold symmetry, and the streptavidin crystal has a twofold symmetry. Yeah? So if the antibody now, with the twofold symmetry, binds to an antigen crystal of threefold symmetry, so let's consider the first FAB arm binds, but the second one cannot bind ideally, yeah? Because the symmetry doesn't fit. So this means that the arm is distorted. This distortion causes a mechanical stress, which makes the FAB arm dissociate. Chemical affinity forces to bind back, but not necessarily to the same antigen, maybe to another antigen. Something like this. And this makes the antibody walk. And here, in the case of the strep diamidine crystal, we have twofold symmetry, but there's also a rotational angle still, even if there's twofold symmetries. But there's many possibilities to bind in twofold symmetry, but it still depends on there's still a rotational freedom between the epitope of the antigen and the paratope of the antibody. And this is why only one direction leads to perfect binding. Yeah? And this is why the orientation of these antibodies is all the same. I mean, we could also analyze this walking process. That is something which is known from nanoscopy. So you analyze the mean square displacement, and if this is linear, like it is shown here, then it is a random movement, like Brownian motions, like diffusion of lipids in membranes. But the diffusion that we see here is much smaller, because you can see that on purple membranes we have a diffusion constant of 6 square nanometers per second. Diffusion of lipids in membranes is 1 square micron per second, so much smaller. And on the S layer we have like 200 square nanometer per second. And there's some characteristic lifetimes. The lifetime of a FAP armor on a purple membrane is 0.5 seconds, and the lifetime of an antibody on the S layer is 0.04 seconds. Why is the movement on the S layer so much faster? That has to do because also the crystal size is larger for the S layer. Yeah? So this means that the mechanical tension on the antibodies is larger on the S layer than on the purple membrane. Okay, and actually the consequence of this movement of the antibodies is that they can cluster easier. Yeah? And this clustering is important because the next step in the immune cascade, I mean the first step is always binding of antibody to antigen in the immune cascade, but the next step is a binding of the complement protein. Yeah? And the complement protein C1q is a hexamer. So it's advantageous if the antibodies are arranged in hexamers. And this is eased by the walking of the antibodies. I mean, there's actually many now also follow-up studies of that. Yeah?
What's dwell time in T-lag?
Pardon me?
In the previous graph, in the previous slide.
Yeah?
What's dwell time and T-lag in the x-axis? The last three graphs. Yeah.
Here.
What is dwell time and T-lag?
Yeah. So the dwell time is how long the antibodies stay on one place. Okay? And that's just a histogram. So you can see that here, and this gives you also, this is the cumulative fitting of this curve, and this gives basically an average dwell time of 0.5 seconds. Yeah? That's how long one antibody stays on one place. This is for the purple membrane, and for the S layer, the dwell time is 0.04 seconds. So short that dwell time in the faster walking.
And the T-lag? In the last.
T-lag, yeah, that is from the mean square displacement, because what you measure is basically you average over the so you have a delta t basically. So what you make is you make a tracing. Okay, so you plot all the data, and then you plot how far it moves within a certain delta t, and the delta t is the T-lag.
Okay. Thank you.
Sure. Yeah, and this research went on further, where actually also together with the antibody company in Netherlands, and where we can also image the aggregate size of the antibodies, so monomer, dimer, trimer, tetramer, pentamer, and hexamer. We can see that here, and so this is how the association. Actually, the driving force of the antibody association is in the FC region. Yeah? And we also were working with mutants that had mutations in the FC region, and this influences everything. But then finally, also we imaged the binding of the complement protein to these hexamers, and we could see that the complement protein has, of course, a much higher affinity to hexamers than to a single antibody. Yeah, then just some other examples there from our lab. So ribosome stalk dynamics, for instance, that we, of course, now we are talking about movies, yeah, with this time resolution, and or for instance, how the integrin bends and unbends on surfaces. This is everything that can be filmed. Or also GROEL, GROES. This is kind of association of GROEL to the GROES, which is driven by ATP, and what we can use here is a caged ATP. So we can, if we uncage it, everything becomes active, and the GROEL can bind to the GROES. Okay. Then also membrane proteins. This is a aquaporin channel, and we can film the conformational changes, yeah, of this aquaporin channel, and can from, yeah, and we can depict, basically we can create an energy diagram from there, okay, so just from the thermal motion, but you can literally film conformational changes of membrane proteins with the high-speed AFM. Yeah, and it has to do with the loop stiffness, so you can assign this movement to the structure of the protein, and it has to do with the loop stiffness of the internal configuration of the membrane protein. Or what we also did, we created suspended membranes and bound streptavidin to it, and we can also follow the diffusion of proteins in the membrane with this technique. Okay? So you can see this tracing the lateral mobility of proteins on membranes. Okay, yeah, and this is it is an old presentation, I should also say, from the. So I mean, this literature is just some kind of older examples. Now, many papers have been published with the high-speed AFM since then. Yeah. Okay, so that is now about topography and recognition imaging. The method was developed in our lab, and also then commercialized a PicoTrack module through Keysight. And if Keysight wouldn't have put down the AFM business, you could still buy it there, but unfortunately not anymore. So I want to talk about about some examples and also some physical aspects, but I decided not to do the math here. Yeah, so I will just show you the implications. If you're interested, we also calculated everything. If you're interested in the math, then please follow up this paper. So how it works. We oscillate the cantilever carrying a ligand, and we are actually using the Mach mode for it. Mach mode is a mode where the cantilevers are magnetically coated, and you drive the cantilever directly into in a magnetic field. Yeah, so you don't oscillate the whole chip. You just oscillate the cantilever. And it also works with tapping mode track imaging, but you just get some kind of cleaner signals from the Mach mode. And what we do is, you can see a trace here. So if we record just the oscillation, so we scan, we perform a line scan. This is single molecules on mica. I think it's lysozyme molecules, and we oscillate there, yeah, and look how the oscillations are influenced by the proteins. And you can see that when we hit the protein, it reduces the downstrokes, yeah? But it doesn't have any influence on the upstrokes, okay? This is what we do with the bear teeth.
So this disabling slow scan means you're taking line
Yeah, right,
Same object.
right, yeah, right. That is just a line scan, and that's why you see the same here, exactly. This is with a pair tip. Yeah. If, pardon me,
The proteins, why they are arranged in a single line?
because we deactivate the scan into this direction.
Going on the same protein.
It's always on the same
Line
same.
is plotted just next. That's what I wanted.
Okay.
Yeah.
That was
So we deactivate the scanning in the y-axis. It always scans on the same. Okay.
Okay. Okay.
Usually it moves one line more up during scanning, but here we deactivate the y scanning axis, and that's why it always scans the same line. Okay.
No. So
This is the particles. They are scanning it again and again, and then
while plotting, we plot the next line.
mhm.
So you go over it again, but that line is plotted next. So it's just the same thing again and again.
Okay.
consider those are multiple particles.
Okay.
Yeah. And if we compress the oscillation, so this is with the pair tip. If we scan now with the tip that carries a ligand, you can see that also the upper strokes are influenced because the ligand binds and reduces the upstrokes. Okay. But what is important is that upstrokes and downstrokes are independent from each other. Yeah. So here again, with the pair tip, we only see information on the bottom, not on the top. With an antibody tip, antibody against lysozyme, we see information in the bottoms and also in the tops. Bottoms is from the topography, and tops is from recognition. Okay? And if we block the tip with lysozyme, that's the proof of specificity. We retain the downstrokes, but the upstrokes disappear. Okay, that's the proof of specificity. Okay, so what we are then do, because upstrokes and downstrokes are independent from each other, we can separate them and use the downstrokes. We feed the downstrokes into the AFM controller, into the feedback loop, and get a topography image. Yeah? So feedback loop, we just servo on the downstrokes and not on the full amplitude as in normal tapping mode. And we detect the peaks from the recognition image. Yeah? This shows us where so we can localize binding events with this. Okay? So that's the video how it works. So we have the ligand, and topography, we always get information from the downstrokes. And then when the ligand binds to the protein, we also get information in the recognition image. Okay? But what you can see, we don't dissociate during oscillation. Yeah, we keep the oscillation amplitude small. We don't dissociate. We just dissociate laterally. Okay? I will come to that in more detail. Okay, so this is normal mag mode imaging. So like 8 minutes. Now we can also do it faster. Topography image, avidine now on mica, and with the biotinylated tip, we can detect binding of biotines, binding of biotin on the tip to the avidine molecules. These are our early applications. So also on histones, for instance, where we also used antibody to detect the histones on the surface. So that is our kind of first publications with track imaging. And later on, we're also using it on cells. Yeah, so we can. This is cadherin-cadherin interaction. So the cells contain cadherin, these adhesion molecules. Yeah, they provide adhesions between cells. And we put cadherin molecule on the tip and sends the cadherin molecules on the cellular surface. We can see these domains of cadherin domains. Cadherin interaction requires calcium, and if we chelate the calcium with EDTA, you can see that topography image remains the same, but the recognition image disappears. Yeah? Then we can overlay topography and recognition images and can map receptor domains on cells in this way. So the domains are 30 nanometers, but we can also image single cadherin molecules with this method. Yeah. Okay. Then I come a little bit to the details of the imaging techniques with avidin biotin. Yeah, so we have biotin on the tip and avidin molecules. So that's all avidin biotin in the next slides. Okay. So this is when we. This is normal MEC mode. Yeah, when we servo on the whole amplitude. Yeah. So you can see it lifts because it holds the amplitude is constant. There's some delay, and this is the topography image because the cantilever gets lifted. Okay. So this is normal tapping or MEC mode, where we servo on the full amplitude. If we have now a biotin on the tip, and if we servo on the whole amplitude, okay. So this again with the bare cantilevers, we get the right height of the protein, okay, when we image. But if we have now a biotin on the tip, we overestimate the height of the protein. Why? Because we don't only get signal from the protein, but also from the binding of the biotin. That both reduces the amplitude, yeah, and so we overestimate this amplitude reduction, and this is why the feedback goes too far up. Okay, so this is why we servo with the half amplitude, so just with the downstrokes, and if we just servo with downstroke, we get the right amplitude, but also a recognition signal. Okay, so basically this is why it's much better to run the feedback loop just with the downstrokes and not with the whole amplitude. Okay? And that's what we can also see here. So this is if we now compare half amplitude with full amplitude feedback, we can see that with the full amplitude, we overestimate the height of the protein, 4 nanometer instead of 3 nanometers. But of course, we also get a higher recognition signal. Why? Because the scanner is further away from the surface. It stretches the crosslinker more. Okay, so this would be an advantage. You get a better recognition signal if you put your feedback on the whole amplitude, but you overestimate the height of the protein. Yeah. So that's why ideally the feedback should just be put on the downstrokes. Then it's also important to choose the right amplitude. This is the right amplitude. Yeah? This is the right amplitude, where you see both topography and recognition signals. Oops. Sorry. So again, this is the ideal amplitude. Here, the amplitude is too small, yeah. So you don't get a signal from stretching of the crosslinker, yeah. And here, the amplitude is too big; it always dissociates, yeah. And then the consequence is because we are oscillating so fast, and if we detach the bond, then the probability to get it in the next oscillation is rather low. Yeah, because we are oscillating so fast. Yeah? So basically, we really have to choose the right amplitude. Too small, we don't stretch the linker, we don't get recognition signal. Too large, we dissociate, and then the probability of unbinding is very low. That's also what we can see here. So here, if we vary the amplitude, so with 14 nanometers, we get a nice recognition image. With 11 nanometers, not so much. Still a little bit with 11 nanometers. 14 again, very nice. 17, we don't really get a recognition image. Yeah, the amplitude is too large. 16 is still fine. 12 is a little bit too low. 14 is kind of the ideal one. 18 is also a little bit. 20 is too high, and here 14, and we get it back. Yeah? So we can really kind of control, and that's also a nice control that it's not unspecific tip surface adhesion. Yeah? We kind of call it amplitude block, yeah, because it's just a window where it works.
So the interlink correlates with the kind of amplitude.
It correlates, yeah. So exactly, yeah. So if you use a shorter linker, then you have another ideal amplitude. Yeah. Okay, then of course, block experiment. This is where we inject avidin into solution. Then we also lose the information about the recognition image. Okay. The other thing is that the frequency is important. Okay. If you push on a spring just one time, you induce some amplitude, and the amplitude decays exponentially. Okay, that's with all harmonic oscillators, and this is what you see here. But this is what we do when we hit the protein, of course. Yeah, when we hit the protein, we get an impulse. Yeah, and this impulse is then exponentially lost over a certain time, and the characteristic time for it is the Q factor of the cantilever. Yeah, so. And let's remember, we kind of the delta T between topography and recognition detection is a half oscillation period. Okay? Yeah? So here we have topography. We detect topography with the downstrokes. Yeah? We excite the cantilever, and the information gets lost exponentially. Okay? But here we detect the recognition. Yeah? And what we see here is that the information is not completely lost when we detect the recognition. This means we have a crosstalk between the topography and the recognition channel. If we choose this amplitude. If we choose this frequency. Yeah? If we choose a smaller frequency, this should be fine. Because here, topography, recognition, and here the information is lost. Okay, so there is no crosstalk between topography and recognition image, and this is what we can test. Yeah? As I said, we did also the math of it. You can follow it in this paper, but that's what you can see. So here's the topography image. We servo on the half amplitude. And this is the amplitude error image. Yeah? So this is basically the difference due to the slowness of the feedback loop. Yeah? So this is basically the information what we have at delta T0. Yeah? And this is then the information of this error image, what we still have after a half oscillation period, yeah? Because this is a recognition image. Okay?
Necessarily have to sit on resonance?
Yeah, you have to be close to resonance, otherwise the signal is very small.
Then you are like halfway down at 0 to 18
Yeah, there you still get a signal. Yeah, yeah, because I mean the resonance peak is very nice. I mean, of course, it's also broad, yeah, but because you directly excite the cantilever, it really looks like a damped harmonic oscillator, and that's why you can so you still get sufficient signal with half of the resonance frequency. But we can see that here, so the higher the frequency, the larger is the crosstalk. Okay, yeah, because this is what is basically at delta T0, where the feedback loop cannot follow. This is how the cantilever is excited. And in the recognition channel, you see how much of this signal in the error image is lost after half oscillation period. Yeah? And okay, so we better do these experiments, let's say, with kind of 9 kilohertz. That's already good. There we almost have no crosstalk from the topography channel. Okay. Okay, so it is really important to have half amplitude feedback, that you get the right topography image with track, yeah. Because I mean, for normal tapping mode, not, yeah, because there you don't, if you don't have functionalized your cantilever, yeah, then you don't have information in the recognition channel, yeah. So with the bare cantilever, it's fine to servo on the whole amplitude, but if your cantilever is coated with a ligand and the ligand binds, then it's much better to just servo on the downstrokes, okay, to get the right topography information. Amplitude has to be properly chosen. If it's too small, then we don't have mechanical response from stretching the linker. If it's too large, then we always dissociate, and the probability for rebinding is very low. And also the driving frequency has to be accordingly chosen in order to minimize the crosstalk between topography and recognition image. Okay. Then also just some examples for track imaging. This is on a bacterial S layer, and so that's the outer membrane layer which can be recrystallized on surfaces. And here, each seventh protein carries a strap tag. Yeah, so this is the wild type, and each seventh protein here in this membrane carries a strap tag, so a recognition tag. And if we bind strapped avidin to the surface, we can see that we only detect in 1/7 of the protein a recognition signal. Okay, yeah. So this is because to the wild type protein it does not bind, only to each seventh, and we can localize this. This is the overlay of the topography and recognition image, and that's the model of it, what we get. And the positional accuracy for detecting a recognition site is 1.5 nanometers. And on the same template, we can also bind gold particles. This is also because gold binds to cysteine. So this protein carries two recognition tabs, one the strap tag where the strapped avidin on the tip binds, and the cysteine where gold particles can bind. And we can see that here if 1/7 of the proteins carries a cysteine, we see it sparsely covered. If 100%, we see a nice template of gold particles. Okay. All right. Yeah.
So Peter, you mentioned about the frequency. So if we suppose use a high frequency and do the measurements at off resonance, say like how peak force tapping does, can we avoid these crosstalks better?
If we do it at high frequency.
And then we do, the cantilever resonance is at a higher frequency, and we do the measurements at off resonance, like how it happens in peak force tapping. Will it be better?
Yeah, sure. Yeah, the slower the amplitude, the less chance for crosstalk. That's general. And if we do it, for peak force, but I mean, in peak force, that's another technique, because there you dissociate always, right?
Yeah.
Yeah.
Oh, so you don't want to dissociate this?
No, in track imaging, I mean, of course, the advantage in peak force is that you can measure forces, yeah, which you cannot do with track imaging. Yeah? But our philosophy is somehow that we first identify a protein with track imaging. We identify the localization, and then we stop there. I mean, there's publications also about it, and then we do force spectroscopy there on this, because the track is something for quickly finding a recognition site. And for spectroscopy, you need to do statistics anyway. And track imaging is always faster than peak force, because it's a resonance technique. Mm-hmm. And peak force is a non-resonance technique. So with each cantilever, track imaging will be faster than peak force.
I see. I was just wondering because the Q factor was very low, about 1, so I thought maybe doing it off resonance. But yes, your point I understood. Also, another thing is, you said that we don't dissociate, but the bonds break laterally, like when the tip scans, right? So does that lateral stretching while it is scanning give some artifacts to the lateral dimension of your molecule that you are imaging?
Not really, yeah, because it is pretty symmetric, the recognition spot, yeah, and if it would influence it a lot, because on the other hand, it can also already bind before it reaches the site, because the link is flexible. Yeah, so what you might scan too fast to dissociate, this is also already when you approach it that it binds before the very tip end reaches the protein. So the recognition spots appear to be really quite symmetric.
Thank you.
Recognition spot will be shifted by some compared to overlay the recognition spot will go little further because now the linker is stretched and really recognize and really change in pop.
Yeah, I mean theoretically you're right, yeah, because even if it can bind before, yeah, it's not in a stretch position, and so yeah, but we analyzed it and we see the recognition spots they're changing a little bit because it's again everything is probabilistic, but we couldn't identify a symmetry into scanning direction. This is not what we see.
Also, one more thing, like, have you compared or correlated this recognition through amplitude with the phase data that you get here? Do you see any recognition through phase? No?
This was our first approach, but it seems to be too much of a damped
Yeah, Q is
harmonic
low.
oscillator that we get a significant phase signal, and the linker is too soft. So that was an approach where we failed, yeah, because this is what we tried before track imaging.
Thank you.
So,
So, what kind of challenges one can face while doing high-speed AFM with the molecules like myosin? Because to me, it seems simpler than doing a fluorescence imaging by modifying the proteins, like attaching a fluorescent tag and then taking care of those fluorescence that it won't go away. We need to add different other agents like anti-fade agents that can affect our protein. So how easy, or I mean, I wanted to understand the difference. Is it simpler to do this, and what kind of challenges you face when you do?
when you do high-speed AFM, well, I mean, I used to do fluorescence microscopy before, yeah, but not nanoscopy. I couldn't say what is easier, what is more. I mean, I think it's always if you can handle the microscope, then maybe sample preparation is but this is general for AFM, not only for high-speed AFM. I would not say that operating in high-speed AFM is more difficult than operating another AFM. Maybe this gives you the answer. If I would compare it with single molecule optical nanoscopy, I think it's the same. Also, if you have the technique in hand, it's also sample preparation. So
Because here the sample preparation is just adding our proteins or attaching our proteins onto the slide and start imaging.
yeah, but that's also what you have to do with fluorescence microscopy.
But initially we will have to modify it, like we have to attach probes and all. Yeah.
But that is also not the big advantage to fluorescence mark a protein. So I would think it's pretty comparable.
Okay. Yeah. Thank you.
Main advantage I see is level free, which means sometimes the tag itself is bigger or same size of the proteins, and then that probably is one advantage.
Yeah.
Tagging a protein affects the activity of
And also activity.
Yeah, depending on, but if you site specific fluorescence label does not really destroy the function. No, I mean the advantage of the AFM is certainly that the resolution is higher. Yeah? I mean, of course, you have in optical nanoscopy, I mean, we have to distinguish between resolution and positional accuracy, right? And in optical nanoscopy, the resolution is still not better than 200 nanometers. So it's just the positional accuracy, and here you have clearly a better resolution. This is what I would see is a advantage of AFM. And the disadvantage in being slower, I mean, sure, it's still slower because, but maybe 5 milliseconds. Okay, you can maybe in 100 microseconds you can get an fluorescence image, and so time resolution is still faster with optics. Because even if it reaches 5 milliseconds, I think if you do like single dye tracing, I think you can go down to 100 microseconds. That is still a factor of 50 in the time resolution, but then it's a factor of 50 better for AFM in the spatial resolution. Yeah. So okay, that's a trade-off, the factor of 50 between time and spatial. Exactly. Yeah, that's right.
Low integration time also will drop the resolution. I mean, you would not get at least the walking and all. You need to collect long enough. Frozen signals.
For you mean in fluorescence now or?
And then nanoscale, optical nanoscale.
Yeah. Of course, it's not if you want to trace movement. Yeah, but sure, but
Fluorescence coming from single molecule.
but I'm just talking from one single image, yeah, and that is 100 microseconds optical microscopy, and now let's say 20 milliseconds or whatever. So and this makes a difference because of course you also have to record many AFM images to see the movement. But one image you're faster with optical microscopy than still with high-speed AFM.
Yeah. Peter, so while doing high-speed AFM imaging of structures like, for example, a single-strand DNA on mica, which can just move, so how do we avoid these things to get a better resolution? Because these things will be always moving. Right?
Yeah, depending on what you want to see. I mean, if you want to see movement, then maybe the art is to attach it to a surface that you can do AFM imaging, because we can only do it on surfaces, but to keep it still mobile. So I would think actually it's an advantage because then you can see that if you image a moving structure with a conventional AFM, then you just see it blurred. Yeah. And with the high-speed AFM, you have a much better chance to get a sharp image because you image faster and even detect the movement. That's how I would see it.
I see. And also, even with high-speed AFM, have people used the phase channel, or do they get it or no because of the low Q factors?
No, the Q factor is not so low for the. This is also around 1. Yeah.
So, but do they get phase data then?
We never did it. It's certainly there, the phase image. I'm not now aware of some spectacular application of phase imaging in the high-speed AFM, but I'm sure that there are some papers out there with. So the phase channel is obviously there. Yeah.
I see. Okay. Thank you.
You can certainly also do this material contrast type of thing. One thing could be the oscillation amplitude is very small for the high-speed AFM. Yeah? So it's just 1 nanometer, and amplitude reduction is 1 to 2 angstrom. Yeah? And I could imagine that if you want to detect like material properties with the phase channel, that maybe the signal is a little bit lower because you just push into it very gently. This is what I could imagine. Yeah, but yeah.
Because there is some work done or dissipation at the interface, so that dissipation could be more sensitive.
Yeah, I mean, of course, you're much faster, so then this would mean if the velocity is higher, this could then also lead to a higher dissipation.
Yeah, that's true.
Yeah, but and I didn't really see it coincidentally in the literature, something spectacular there, but it might well be out there. So.